Hi, this is Miss Litton, and this is my third period AP Bio class. Say hi.
Hi.
And this is our introductory lab, lab 1 with AMGEN. So a couple of things we're going to go over quickly is an overview of today's lab. We're going to talk about your collaborative hyperdoc, and then you're going to go through and complete lab 1. And then if time, you're going to start preparing for lab 2. The point of today is to learn how to use the micropipetters to move small amounts of fluid and to get very comfortable with that, because today you're going to be working with dyes and you'll have a lot of them with a lot of fluid in the tubes and easy to see. But when we do the actual lab, most of the time you'll bring up a microfuge tube and you'll say to me, Miss Litton, where's the stuff? And I'll be, it's right there at the bottom, because there will only be 10 microliters of fluid in there and it'll be clear. And the thing about this lab is that once you've pulled up a solution or mixed a solution, there's nothing I can do to fix it if you've done it wrong. And you can't tell what's in a tube once you've removed it. And so you have to be really careful with A, setting your micropipetters accurately, B, making sure that you can actually see the fluid at the end of the tip of the micropipetter, 3, making sure you dispense all of that fluid into that container and verifying it. The second part of today is to practice doing gel electrophoresis. Now, for us in our lab, gel electrophoresis is going to sort our DNA according to size. And so we will be comparing cut and uncut DNA, and we will look to see at ligated DNA, and we will compare that against a lambda BC marker. We use mini ones, so each one of you will have, the cool thing about mini ones is you can run your lab in about 20 minutes when you're separating your DNA during class and look at your results and analyze it. Today, when you run your dyes for practice, you only have to spend about 15 minutes, and then all your dyes should separate out that you're practicing with. So I'm going to walk through how to set that up as well. Okay? So the first thing I want to talk to you about is the micropipetter, and you already should have somewhat of a working knowledge with the micropipetter if you watch the videos that I asked you to look at. What is common about all micropipetters is they have a first stop and they have a second stop. So I know you can't see this at home, I'm sorry. Watch the videos on this part. But when you're holding it in your hand, you will be able to feel a resistance. That is your first stop. That is a measured amount of fluid. But yet I can go to a second stop, and that's like a little bit of bonus air to help push fluid out. As far as adjusting them, it's this part right here on top. This is where you turn in the amount that you want. So when I look at this right now, what does it say?
9.3.
9.3. So yeah, it does. Okay, so it says 9.3, and that's easy because it has a decimal on it for you to be able to tell. That means if I push it down to the first stop, I am pushing out 9.3 microliters of air. I can then put this into a small tube, slowly lift my thumb up like this, and suck up 9.3 microliters of fluid. When I want to go dispense, I push down. That makes sense, just like a dropper, but then you floor it, and it has a little bit of bonus air that goes push and gets all the rest of it out of the end of the tip. Okay? Now, next thing we need to talk about is the end of the micropipetter. If you were just putting in a bunch of different solutions, I kind of introduced this to you before, this area would get what? Contaminated. So what do we need to do?
Tip.
Put on a tip. So that's your life lesson. Never dip without a fresh tip. And in your box, you each have a box that looks, I know, and you have a box that looks like this. Okay, and what you're going to want to do is this will be sitting on your table. Only open the box when you're getting a tip, otherwise close it. I'm going to tell you it's a real bummer if all of these fall out. Okay. Also, whatever tips you use today, use as many as you need. At the very end of the lab, you're going to need to have somebody wash their hands, and in the back of the room right there, you can restock the tips into this box so they're easy to access. To put a tip on, you just gently push firmly because you want it on there, but not so hard that you break it, and then close this back up. If you want to ensure that you have it on there, if you grab towards the top of it, then you can tell that it's not wiggling. Okay, and then you go into your solution, etc. Now, if I have this set at 9.3, now I'm going to bring it down and watch as I roll it down. Now I'm at what? 4.3. Do you see the difference in how this moved? Okay, so that is your indicator where this is. That part should naturally change if you want to pick up some fluid. So So I want to model for you how to pick up fluid. Okay, so I put on a fresh tip. I don't lay it down on the counter because then this would become contaminated, right? So I'm like, oh my gosh, I've contaminated this one. How do I get the tip off of here? Eject. Now notice this is the way you hold a micropipetter. You don't get to hold it any other way. If you're left-handed, right-handed, it works both ways, okay? But your thumb goes here on top. Only when you're ejecting, then you go right here, and it'll push it off so you don't need to touch it, okay? So I want to make sure it's at 10. Is it at 10? 10.0, so I'm good. So I'm going to get a fresh tip. Okay, and you have in the back of the room, you have the things that I need to give you that day, as well as you will have a box that looks like this. These are your stock tubes to use. And that means this case right here only has my stock tubes in it. If you make any tubes, which you will each be making your own microfuge tube today, do not put them in here. You put those in your own microfuge rack, okay? So in your stock box today, when you lift this off, you will see in there will be a color of red dye, okay? There will be some distilled water, and then you have tubes labeled 1, 2, 3. These 3 different dyes are going to be going into your gel today. So the only thing that's going to go into your gel today are the tubes labeled 1, 2, and 3, okay? But you will use the water and the red dye to create your own tubes when you're in your groups, okay? So I'm going to pick up the red dye here, and let's say I wanted to have 10 microliters of fluid. Okay, so I set it at 10. I open this up. Every time you use the micropipetter, everything is at eye level. Okay, everybody's paying attention up here. Everything's at eye level. You don't put it down and leave it in the rack and pull it out. You don't have a friend hold it for you. If you're taking the solution, it's in your hands and it's at eye level. What do I need to do first? Yeah, so I need to push it down to the first stop. Okay, so I'm going to push it down to the first stop. Then I need to see that the tip is actually in the fluid. Today, it's going to be easy peasy. I'm just going to tell you tomorrow or next time, Monday, it's not, because it's going to be down there at the bottom. Okay, and then I slowly release, and now I have 10 microliters of fluid at the end of my micropipette. Keep in mind, 1 microliter of fluid is 1/1,000,000th of a liter, right? So you might want to get comfortable with what each amount of the drops look like. That's why I have these. It's not part of your lab today, but if you want to just do it for comparison's sake, I have this plastic sheet, and it has where you can put a 20 microliter drop, a 15, a 10, a 5, or a 2. Totally up to you. I'm just putting that in here as something you might want. Now, back here is where you will get your tubes. Okay? And all tubes on them have a shadowy region on them. Can you see the shadowy region?
Yeah.
And that's where you label all your tubes. You can also label them on the cap up top, but I also want you to label them on the side. Always label them on the vertical because you hold a tube vertically when you remove fluid, and you don't want to have it written sideways where you go like this. Does that make sense?
Yeah.
Always, and you write heavy and dark with a Sharpie, and you have a Sharpie in your box as well. Okay, so let's say I want to put this 10 microliters in the tube. What do I need to do?
Push down slowly.
Yeah, so and the best micropipetters put their solutions right on the bottom side of the tube if you're going into a tube, so you can actually see that the drop is going in there. Now, where do I go? All the way to the second stop when I'm dispensing. Then I need to remember to pull my hand out before I let go, right? So now I know that I have a 10 microliter drop right there on the side. If I had to add a different solution in, then I would have another drop on the side. But I need those to be all together, right? So I need to condense them. Or let's say I accidentally drop my tube. Where'd it go? Oh, it's right there. No, no. Okay, so now I have drops all over the place. Or let's say I put in 3 different solutions. Now I need to condense them. So what you use to, where's my, oh, there we go. So what you use to condense them is you each will have your own personal private microcentrifuge at your lab station as well. The microcentrifuge, to open it, you just pop the lid like this. Okay, if I only have 1 tube I need to spin, I need to go and get a blank tube because I need to be able to have it be balanced when it's spinning. Okay, then you just close it. Of course, yours will be on your desk. There's an on switch right here in the back. It'll start to spin it, maybe 10 seconds or so. not 10 seconds, like 5 seconds. I don't know what I'm saying. 20 minutes. Okay, when it stops, then you're able to open it. And now you can see that all those drops are right there at the bottom. Now, technique, you want to get this down right now. Can you set this at 5? Okay. Not only did all the drops go down there at the bottom, but when I was spinning it, then if I, let's say, had any plasmids or any dye particles, they not only went down like this, but they could be at the very bottom of the tube. So when I go to remove 5 microliters out of here, I may not be bringing out, could just be part of the dilution, not actually the stuff I want. So whenever you, are we at 5? Agree? Okay, see how I'm asking every time? So whenever you want to remove a solution out, let's say I want to have 5 microliters out of this 10 microliter tube, then I open it up, okay, I go down to, I set it at 5, that's how much I'm going to remove, but I don't remove it right away. I go into the solution, put my tip down in there, and then I just pump it a couple times. By doing that, what am I doing? I'm mixing it all up, and then I can go in there and remove 5 microliters out. Does that make sense? Now, what happens if when I go to remove it, okay, what happens if when I go in to remove it, I do it like this, where I get air bubbles in it like that? Is that a good draw? No. What should I do? Throw this away and eject it and go in again? No. Everything is accurately measured. I'm not kidding you. I'm giving you 10 microliters of very expensive plasmid. You need to pull 5 out and put in one tube and 5 out and put in another tube. Is there any left over? No. You'll probably have to spin it between draws because you won't even see it. Okay? So you cannot just eject this. What should I do? Put it back in there. Exactly. Put it back in there. Get it all out, and then go again. Make sure my tip chases the volume. Oh, didn't get it in there. Make sure my tip chases the volume all the way down so that I have it in there. Does that make sense to you? Okay? So the first thing I want you to do is when you get these is just to mess around with them and feel for the first and the second stop. Now this, you have 3 micropipetters available to you. This one is called a P10, so it only goes up to 10 microliters. It's easy peasy, has a decimal. So if it says 5.7, it's 5.7 microliters. This micropipette is if you look at the top, it says water, and then what does it say?
2 to 20.
2 to 20 microliters. Now it has no decimals, but if it says 180, since you know it only goes up to 20 microliters, right? It's a 2 to 20. If it says 180, what do you think that means? 18.0. Or if it says 0 to 5, that means 2.5 microliters. Does that make sense? It's where, and it's hashtagged on the bottom. The third micropipetter, and you won't need to use this one today. This one is a, look right here, what is this one? 20 to 200. This one will not accurately measure any tenths of any milliliters because it doesn't even start measuring until 20 microliters, right? So for this one, if it says 25, it means 25. If it says 175, it means 175. Follow me on that? So you need to accurately look at the tool that you're using, what it measures from before you start using it, because I've had people make major mistakes because they thought they were doing, 17.5 and they were doing 175, and it kind of wrecked it for them, okay? All right, so we won't let that happen. Your rack to hold your tubes is right here. So anything that you make today goes into your rack of tubes, okay? And then you also have your Sharpie in here, and then your tips, and then your trash. And sorry, I'm making yours messy, Team 6. Also at your station, actually, real quick, in your own words, oldest bio buddy, explain how to use a micropipette. Go ahead.
Right, so you have your first and second tube, but at the first stop, you put it in, release, then you put in the other one, push all the way down, and then one that you can
All right, and then come back to me now. So, here we go. So, today you'll be making you'll make a tube for practice. You can practice on that. And then the big thing that you're going to do today is do gel electrophoresis. So, I'm excited for you that you have your own gel box that you can use, and I want to make sure that you understand how to work it right now. So I'm going to show you a video that they use it to show to teachers to learn how to do it.
This is the Mini 1 gel system. It comes with a hood, gel casting trays, combs, an electrophoresis chamber, also known as the buffer tank, a black plastic indicator, a cord, and a power supply that is connected to the base. The cord connects to the power supply here. Please remember that you cannot pour buffer in the base. Buffer must be poured in the electrophoresis chamber because there are 2 electrodes that are within the base that may get destroyed. In the buffer tank, there are 2 little bumps on one side. These are the bumps for the electrical connection. The other side is completely flat. This is an indication on how the buffer tank should be placed into the base. The buffer tank should fit snugly into the base. Force should not be needed to fit the buffer tank into the base. Inside the buffer tank, there are 2 rods. These are the electrodes. There is a black plastic indicator that has the markings for the wells. It also has the ruler and positive and negative signs on the opposite side. The positive and negatives should match the positive and negative signs on the buffer tank. There should be an indentation where the black plastic will fit. After making the gels, lay them in and pour in the buffer.
They don't have a gel in there, she's just putting the tray in there, but a gel would be sitting in there.
This is basically the hood and the top. There is an indentation in the middle, and the hood should fit well. After plugging the gel box in, use the power button to turn the gel box on and off. The hood must be on in order to run the gel box. As soon as the hood is removed, the power will turn off. You will know that it is running when the green light is turned on the whole time. Students may play with it and take off the hood. Remind them to replace the top and to turn the gel box back on. Once finished using the gel box, rinse the tank with DI water and place it upside down to dry.
Okay. Hopefully you understand how that works. I'm just going to make sure I put that on. Okay, come back to me. Now, I have already cast your gels for you, and they're waiting for you in the back. I want you to see. What it looks like, and I'm also going to pass around a gel so you have an idea. So the gel is poured into this tray, and then these combs sit in the tray, and then that makes the divots in the gel as it hardens. So I'm going to pass the gel around and just be gentle with it. Try not to break it so that you have an idea of what the gel feels like. It has openings on one side, and you need to keep in mind that when you're doing this lab, you're going to load the gel when it's sitting in the gel box, submerged underneath buffer. okay? So you're gonna have to go through fluid into this well hole and everything's clear, okay? So on one side, I can feel like a little bit of raise from when I pulled the comb out, then you have it like raised a little bit right there. So you're gonna need to get the tip into one of those holes to load it. But the divots go down part way here on the side. You don't want to punch it, right? If it punches and goes underneath it, then it's not going to work. So one side should feel like there's no holes, and on one side there are holes. Okay, so I'm just going to pass that around so you can see that. I want you, if you look, I'm going to show you a video real quick that I just shot with my phone on how to set up the boxes. I want you to see the pieces right here. You already know about the hood, okay? If you can look up here for a minute, that'd be helpful. You can see the power supply and where it goes in, okay? This part right here is the chamber that holds the fluid and the gel. Now, for some of you, there's still solution in your chamber, so you won't have to put this into it because there's already fluid in there. When you load your gel into it, you need to just cover the gel, just barely with fluid. The fluid you need to load into the gel chamber, if gentlemen in the back, if you could grab and just lift it up, it's that clear plastic cap and it has blue and green stripe on it. Yes. Yeah, can you hold that up? That right there is the buffer that you need to put into the chamber. Now, if your box is completely empty, the amount that you need, you have this, all your gel stuff will be in here, unless yours is already sitting on your table. But it'll all be in this green bucket, and this line right here that I've marked, that's exactly how much fluid you need. Now, if you already have your fluid in there, then just put a little bit of extra. Don't take too much of this. Just go get a little bit and just barely cover your gel. And remember, the fluid has to be covering the gel before you actually load the gel. To put this in, because you all have to do this the second time around for sure when you're using your DNA, because you're going to make fresh fluid every single time when you use it. You can see that the two halves are not equal in size, and so that will help you to know which way to put it in here, and then you need to hear it snap into place. This plate right here is removable. It's gray today because you're going to be using dyes, but this plate we're going to change out and make it black when you use the DNA, because it'll help you to, the gray helps you to see the colors, but the black one will help you to see the glowing light of the DNA when you do that, okay? And so here, let me just show you how you kind of snap it all together. Just watch this real quick.
When you put the tank in, there's only one way it can go in. I want you to look to see that your disc that you put in right here is on the far side away from the positive, okay? Then when you bring in the gel, you can see the well holes. You want to make sure they're open to you and line those up with the gray disc here. Goes right in here, and then you just pour the solution over the top. You're going to load on this side, and the DNA or dyes will run towards the positive. You want the solution to barely cover. You can turn on the light so you can see the well holes, and that's it.
Now, if it's running towards the positive, what does that tell you about the charge on both DNA and the dyes? Must be negative, right, if it's running towards the positive. Does that make sense? Okay, and hopefully you did all your background work on that. Now, I want us to agree where well hole 1 is. Right here, can you barely see where it says M for Mini 1? I'll show it to you on here. Do you see where it says Mini 1 right up above the light switches? Okay. The well hole closest to the words Mini 1 is well 1. So it would be 1, 2, 3, 4, 5, 6, working away. So 1 is closest to the words 1. Does that make sense to you? And that well holes need to go right here on this side. I have given you 3 solutions. Solution 1, 2, and 3. I want you to put 1 in well 1, 2 in well 2, right? Are you with me on that? And then repeat it, 1, 2, 3 for 4, 5, and 6. So you'll have 2 runs of it. If there's 3 people in your team, everybody will get to go twice, right? If there's 4 people in your team, then 2 of you will get to go twice. You got that? Okay? And I would make the person, if they stab it or screw up, I would have them go twice so they can get better at it. If you nailed it, you won't need to go twice. All right, from the side, like I mentioned, You're going to be loading here, and you're going to watch this video on things to watch out for when you are loading.
The purpose of this video is to point out concerns when loading samples for horizontal DNA electrophoresis. The first thing to be concerned about is actually the preparation of your DNA sample. You'll want to dilute your DNA sample into a loading buffer. Typically these are found in 10x or 6x formations, and they essentially contain weighting agents, which are typically phycol, glycerol, or sucrose are commonly used. And also they will contain a dye or a couple of dyes that will allow you to monitor the electrophoresis process. It is important that these are diluted properly, otherwise your DNA will actually dissolve into the aqueous buffer versus actually dropping into the well. And that's the importance of the glycerol or the weighting agent. Secondly, it's important to know approximately how much of your sample you will be loading, because you want to make sure that the wells in your gel will hold that amount. If you load more into the well, it will spill over into the other wells and contaminate you and give you possibly false results. The first two examples I want to show you are normal loading. Notice I put the tip down into the well. When I depress the plunger, the sample will drop down to the bottom. As I pull away, you'll also notice a small stream. If this is a worry to you, just hold your tip down there longer so the sample can drop down. Otherwise, it's usually no big deal when you're loading a sample. The next couple examples are your classic air bubbles. The first one is the air bubble at the tip. This is usually no problem. Just before you put it into the buffer or to the electrophoresis buffer, just depress your plunger to get rid of it. The second one is air bubbles within the tip. If you see this, be aware of it before you try to load it. Put it back into your tube, draw it back up without air bubbles. Air bubbles in the tip are going to cause dispersion of your sample and possibly lead to some of the loss of your sample. Here is the classic shoving your tip through the bottom of the gel.
Most common
just
mistake.
Be aware of where your tip is. As you can see down at the bottom, the sample is lost underneath it, and also you have inadequate loading of your sample into the well. Sometimes you can save your sample or have enough in your well to actually do electrophoresis, but other times you kind of just lost some of your sample. Sometimes when you're in a big hurry purifying your DNA, you will leave a little bit of ethanol over your pellet and then dissolve it in your appropriate buffer, usually TE buffer. If there's too much ethanol or isopropanol or some organic solvent, when you go to actually load your gel, you'll notice that your sample will actually float out of the well into the buffer. And so here's this little video showing that. Here's a quick example of what happens when you don't put your tip into the well. As you can see, the weighting agent weighs your material down into the well, but periodically you'll see samples spilling onto the surface. This is usually not a huge deal, but it can contaminate local wells, which could lead to false readings. So just be concerned of where your tip is and try to put it a little bit into the well to ensure proper loading. Last but not least, here's a quick example of what happens when you do not remove the tip before you pull the plunger back up. You'll load some of your sample back into the tip. This actually may not be a big deal, but if you're trying to be quantitative, it may lead to false results.
Okay, got that. That's usually the hardest part, is getting those gels loaded. I want to remind you that the gel box itself is not going to, that's the one time when you're not lifting something up to load it. It stays put the whole time once you set it up. So if you want to make sure that it's close to the power, and then have it set up whatever's comfortable for you, maybe pull the chairs away a little bit, and people will come and load it, and you're not going to move it around once you have the gel in there. And the way I like to load gels is the crane method. So I put both my elbows on the table and then support my hand, my loading hand, and just kind of come down like a crane over the gel. And when you put that tip into the well hole, you can feel that you have a little resistance if you have it actually in the well hole. You'll kind of feel it kind of resist you a little bit, and then you'll know you're in the right spot. Just don't be too anxious and just stab because you're like, oh, I want to get it, you know, or I'll see people, they'll take the top and they'll just be like, you know, because they're so nervous about it. Just slow go and load it in there, and then make sure you're not on the eject button when you do that, okay? So it will stay put. Next, let's go to our hyperdoc and make sure that we know where we're starting on this today. So on your hyperdoc, you should already have completed the first video up on top and answered those questions. You should already have looked into the lab book. Some people like to use an iPad and have the lab book open to that all the time so that you're reading the lab book. My hyperdoc is not the lab book, right? So you're going to need it more and more. This introductory day, I've given you a lot of the information on here, but you're going to need the lab book in order to perform the lab, so you might want to open it up on the right page. A should already be done, B should already be done, so your work today in the lab starts at C. So the first part in C is you're just setting Micropipette Er dials. I'm not looking at the tips, and I'm not looking at the whole micropipette. I'm just looking that you can dial it into the right amount using the different micropipettes. And remember, you have to have a physical watermark that is unique, that no one else would have, that's a part of each one of your pictures. You do not want me to have to zoom in when I look at your pictures. It should be real clear and evident and close in when you put those pictures in, and you have 3 different measurements you need to do. Number 2, I'm sure you have read it, but it says all members of the lab team. So for you on that one, every member is going to pick up a microfuge tube, and you're going to label it with your period and group. So you would be 3- whatever your group is, and put your initials on it. You can label it on the side and on the top, but do also label it on the side. Once you have all your tubes done, you will take a picture with your watermark of everybody's members. Now, this part, task 3, I say begin reading at A24. Okay, I give you the instructions here, but you need the lab book instructions as well. And don't forget, with your watermark, before you turn your gel box on, once you've got all your loads, take a close-up of what the well holes look like. That's what I'm most concerned about, that I can tell where everything is and that you've put it in correctly. And then take a picture of your gel colors once you've run the gel. The gel will take, the lab book doesn't know that you're using mini ones, and the thing about mini ones is they're speedy, which is awesome. So it only takes 15 minutes to run them. So when you're done, you're just gonna take them out of the gel box. You can either flip over that white plastic card I have you for practicing. That's probably the best thing. Take the gel out of the container that holds it. Just take the gel out of it and put it on there, and call me over once you have it sitting on there or on a paper towel, and I'll go over the results with you. And remember, you need to take a picture of it afterwards as well. And then last but not least, 2 things here on task 4. 1, every member and what you did today, and I would do that as you go along. And then you have a Google form. So that there is no confusion, I ask you about what color solution 1 contains. That means after you run the gel, right? Because I've had people lift up those tubes I showed you and they're like, I think it's colors. Okay, the whole reason you're doing the gel is to separate out the colors of the dye. So you answer that question after you've run the gel. Okay, as far as where the gels are, they, here in the back, there are 8 OCD squares set up here. I actually used a ruler. And so here's team 1, 2, they're all labeled. But at team 1, you have this plastic card if you want to do some practicing. Otherwise, you can put your gel on it on the back side. You have your stock tubes that I mentioned. And then in this tray, you can take this out. This is what's going in your gel box, right? And you're going to make sure the well holes, like I made these gels and just flopped them down here. Make sure you can feel that the well holes are open and it's on the side away from the positive, so that once you load it, it'll go towards the positive. Okay? If you need additional solution, remember it's here. Only go to the line if your box is empty. Only get a little bit if it's already got fluid in it. Look to see. You just want the fluid to just barely cover up the gel. And fresh tips are back here and clean tubes are back here. You need to work efficiently. The gel needs to run for 15 minutes, and I need to see it afterwards. So the gel needs to be running by 11:45. That means you have 40 minutes or 39 minutes to take your 3 pictures of your colors, to make your individual tubes, and to load your gel. That is plenty of time. I promise you it's plenty of time. Okay? But you need to be on it and think, okay, are we turning on our gel at 11:45? You can turn it on by 11:30, that's great. You can start working on other things. Clean up. Once your gel is running, throw away all your tubes, restock your tips, right? And pay attention to the way your lab stations look. That's the way I expect them to look when you are done today, okay? Yes, that's exactly how they need to look when you are done today. Are there questions or concerns that you have? Okay, work efficiently as a team. I'm already proud of you. Hope that was helpful.
The watermark, does it have to be like physically attached to our body, or can it be something more fluid?
No, you're going to put it next to whatever item, the gel or the tube. It's going to be next to it in your picture. Okay, have a piece of toast.